fact, in China almost, for example, in rice hybrids, 70 to 80% cultivation is of hybrid, which gives you a lot of production. But in this country we haven't. We have started, a lot of things are going on, but not to the extent that China is. This was the hybrid seed technology. I call it the third revolution, the third development, the development of biotech crops, the genetically modified crops. And the one which has given lots of dividends so far is the DT cotton. India, which once was importing cotton, today is an exporter of cotton because of it. Of course, there could be many views and issues about this. But these are basically the three different ways by which productivity has been increased. Now, biotech crops, how do you produce biotech crops? There are two methodologies. One is marker-assisted breeding technology. You identify the phenotype with a marker and then use those markers in order to breed. It's faster, efficient technology. But so far, not many varieties have been developed through marker breeding. It's a procedure which is still going on. The second method of producing biotech crops is through genetic engineering technology. This is a robust methodology. Both of them are robust, but there has been an increase in the use of this technology. However, this technology is still limited to few traits, few crop plants, and also a few crops. So there is a limitation of this for many reasons. So there is a great potential there to use this technology for improvement of our crop plants. Why we think so that there will be a great potential? Because this technology has now been made possible in many different crop plants, both the normal dicot plants, which are dicot plants like tea and others, but also in the crop plants like rice, wheat, sorghum plants. This technology. And this is a technology which all of you know, most of the students know this is through, I also call it basically breeding manipulation, through the use of a bacterium, agrobacterium, chemotesting, which normally infects the plants. This is a normal process of infection. It's a natural genetic engineering technology where agrobacterium infects the plants and it transfers its DNA into the plant system and develops what we call as a ground. So in agrobacterium, the major DNA is the plasmid DNA. It's the plasmid DNA which is transferred from the bacterium into the plant. So this becomes a vector. And this discovery was made somewhere in the early 80s by a group in Germany and Belgium. And this DI plasmid can now be reconstructed by taking a DNA of a gene from the desired trait, put it into this plasmid here, and this plasmid naturally transfers itself into the plant system from which you can regenerate. This is the whole transformation technology, agrobacterium transformation technology, which all of us are very well aware of. In addition to this, there are different technologies, particle bombardment technology, tech technology, but this is most often used nowadays in plant generally. Now, plant genetic engineering, it's not that you start the experiment today and you will get the result next year. It's a very long drawn procedure by which you can develop a new. This must be understood. So you have a gene of interest which you want to transfer, but this gene of interest has to be regulated by a very specific promoter. And then these constructs through agrobacterium or gene technology are transformed by a methodology which I just described. Right? And after the plants have been transformed, hundreds of these plants have to be validated for the trait for which you are looking. And this is a very important step. You cannot have one transform and start working on it. You have to generate at least hundreds. In fact, some of the groups in America, they said minimum 500. And we know that the GM cotton which India produced, they started working with only 2 units. That's what the problem. So you have to start with large number of transformers and then select and validate. And few of those transgenic events will be validated through generations T1, T2, T3, till you get a homozygous. To do this, you need clearances at the institutional level. And after this, if you want to go for limited field trial, you need further clearance from RCGM, which is working under the Department of Health. Second level, in normal breeding procedures which we conduct, whether it is mutagenesis, breeding and others, no clearance is there. We tell you later on that they also make as much disturbance in the genome as this technology. But there is no clearance. So you get limited field trials. At this time, you have to validate these events, you have to do toxicity tests, safety. And these are what take a lot of time. And finally, you get the GAC clearance to do multi-location trials and the product. Yeah? Which again has to go to many clearances, CDEX and other types of protection. So this technology usually takes 10 to 12 years. And the BT cotton technology about which all of us know, it took roughly around 15 years and $70 million. That's the kind of financial burden as well as the time burden technology. But still people will like to go for it because many of the traits which you want to develop cannot be developed by real. So we need this GM technology in the future to solve the problems in agriculture, which will have to do in the future because of the climate change issue which we are talking about. And these are the problems which we have to handle. Less of water, less of energy, and less of land. This is what we are going to face in years to come. And what will have more of drylands, high saline soils, high CO2, and high temperature. These are the predictions which we have. So the kind of agriculture which we are talking about in the future, the next 20, 30 years, will have to deal with these issues. And can we deal these issues with the current technology, or it would be proper if we also use the new technologies, the biotechnologies, in order to solve the issue. Now this is, if many of you know, that in drought conditions what really happens, the productivity totally vanishes, and agriculture uses 70% of water which is available today, and this really shortage of water affects crop production. And if you overuse water, irrigation systems and other canal systems, you will end up into the problem of salinity. And according to USD estimates, almost half of the world's irrigated agricultural land is going to be sold. So on one hand you have less of water, high irrigation due to salinity. And both these conditions you cannot grow that. So that is the situation. So my question is that the kind of research work which we have been trying to do is, will it be possible to make use of such areas for agriculture with use of biotechnology? Can we ever grow anything in these? Would it be? It's a dream, and that this would be possible. Can it work? We think it may be possible because in the last 20, 30 years, there is a tremendous increase in the knowledge in the field of plant biology. I'm sure many of you will not be following it up, but there are many plant genomes which have been fully sequenced. The initial genome was Arabidopsis, which is a small plant. The whole rice genome has been sequenced, wheat has been sequenced, and many others have been sequenced. So the genomic information of many of the plants is now available to us. And many of these genes have also been validated digitally. Similarly, there is a lot of information on protein-protein interactions, what we call as the system biology approach. We try to understand how plants respond to the environment. Plant pathogen interactions are being studied in great detail. And also, many groups around the world identify, because many of these traits are quantitative traits, they are identifying 2 years for complex agriculture. So because of this full burst of knowledge which has come up in the area of plant biology, the functional genomics which now people are working on, people get confidence that the kind of questions which I ask or the problems which we raise may be possible to take up those. So in our group in ICGB where I was working earlier, we have this a small group of people, we call it a plant stress balance. Now what we are looking at different abiological stresses. Mostly our concentration, my group was on abiological stresses. Within the plant stays low temperature, high temperature, flooding, heavy metals, challenge.